When the four regions are occupied by regulators, they form a complex called enhantiosome. This complex binds with the protein GCN5, an acetyltransferase that adds acetyl groups to the tails of histones H3 and H4. Then a kinase protein also binds to this complex and adds phosphates to the modified H3 histone tails. Once the H3 histones are acetylated and phosphorylated, the GCN5 protein adds an additional acetyl group to them. Once the H3 histones have been modified, the BAF complex binds to them. This complex displaces histones to uncover a sequence called TATA box and activate the gene for interferons. Transcription factor 2D binds to histone H3, and this in turn is bound to TBP, which binds to the TATA sequence in DNA. At the same time, the P300 protein also binds the enhantiosome complex. This protein helps attract RNA polymerase II and transcription factors, so they bind faster to genes and accelerate the reaction. RNA polymerase II begins to polymerize an RNA strand that complements the DNA information. Some proteins add a cap to the RNA on its prime 5 side. Then, different proteins bind to the tail of RNA polymerase II, and these bind to RNA sequences that divide introns from exons. Spliceosome proteins bind to these proteins and separates introns from exons. Finally, other proteins add a repetitive series of adenines, called poly-A tail. At the end, a mature messenger RNA is synthesized and is subsequently transported out of the nucleus to the cell cytoplasm. Here the messenger RNA meets the ribosomes. First, the 40S subunit binds to the mRNA and moves forward until it finds an AUG sequence and binds tightly. Then the 60S subunit binds to it and begins to translate the information from RNA to protein. The ribosome binds to proteins in the membrane of the endoplasmic reticulum and introduces the interferon proteins inside it. The new protein